Genetics and Biochemistry. Introduction to the module. Genetics and biochemistry are very significant subjects for biomedical science. In this module, we will discuss the basic concepts of genes, DNA, genetic mutations, amino acids and proteins. Topics to be covered. Concepts and overview. Genes and the human genome. DNA and RNA. Genetic transmission pattern. Gene mutation. Amino acids, structure and function. Protein, structure and function. Protein synthesis. Concepts and overview. Genetic factors play an essential role in detecting human diseases. Genetics has played a minor role in day-to-day primary care till now. However, recent advancements, especially the sequencing of the human genome, are changing this picture rapidly. It is expected that genetics will play an increasingly central role in all areas of medicine and particularly in primary care. One of the most striking generalizations of biochemistry is that the 22 amino acids and the 4 bases have minor reservations and are the same throughout nature. Proteins are essential for life, so denaturation, breaking of structure of bacterial proteins, results in the death of any microorganism. Genes and the human genome. Beginning with the knowledge that genes work to encode proteins, our knowledge of the gene evolved during the course of the 20th century. The discovery of the structure of DNA in the middle of the 20th century brought in an era of intense research into the processes of gene replication and expression. Methods of DNA sequence analysis were introduced in the latter quarter of the century, culminating with the determination of the human genome sequence right before the turn of the century. DNA and RNA. DNA, deoxyribonucleic acid, consists of a double helical structure, with the 2 sugar phosphate strands linked together by hydrogen bond, between cytosine guanine or adenine thymine bases. The 2 strands are oriented in an antiparallel manner, and the 59 or 39 ribose sites free ends. DNA replication involves a partial unwinding of the strands and assembly of a new strand using the other as a template. Ribonucleic acid, RNA, is an essential biological macromolecule that is present in all bodily cells. It is principally involved in the synthesis of proteins, carrying the messenger instructions from DNA, which contains the genetic instructions required to develop and maintain life. Structure and function of genes. The basic unit of genetic function is the gene, the chemical basis for the DNA molecule. DNA consists of a pair of strands of a sugar phosphate backbone attached to a set of pyrimidine and purine bases. The strands are together by hydrogen bonds between adenine thymine bases and between guanine cytosine bases. Together, these strands form a double helix. The strands separate during DNA replication. DNA, therefore, contains within its structure the information necessary for its replication. The sequence of bases in DNA also provides the code that determines the structure of proteins. Proteins consist of chains of amino acids. The amino acid sequence of a protein is determined by the sequence of bases in the DNA stretch that encodes the protein. Each amino acid is represented in DNA by a triplet of bases, codon. This genetic code is more or less universal to all organisms. One strand of DNA's base sequence is copied into a complementary RNA, mRNA, which translates on the ribosome into protein. The process of copying the DNA sequence of a gene into messenger RNA, mRNA, is referred to as transcription. Gene expression is tightly controlled, with particular genes being turned on or off in specific cells at specific times in development or response to physiologic signals. All amino acids have the same basic structure. At the center of each amino acid, there is a carbon called the alpha carbon, and it is attached to 4 groups: hydrogen, an alpha carboxyl group, an alpha amine group, and an R group, sometimes referred to as a single chain. The alpha carbon, carboxyl, and amino groups are common to all amino acids, so the R group is the unique feature in each amino acid. 22 amino acids are found in proteins, but the universal genetic code specifies only 20. Selenocysteine and pyrrolysine use tRNAs that can pair with stop codons in the mRNA during translation. When this happens, the unusual amino acids can be incorporated into proteins. Essential and non-essential amino acids. Enzymes contain selenocysteine, including glutathione peroxidases, thioredoxin reductases, formate dehydrogenases, glycine reductases, and selenophosphate synthetase. Nutritionists divide amino acids into two groups. Essential amino acids must be in the diet because cells cannot synthesize them. Non-essential amino acids can be made by cells. Essential amino acids vary considerably from one organism to another and even differ in humans, depending on whether they are adults or children. Some amino acids that are usually non-essential may need to be obtained from the diet in some instances, especially for individuals who do not synthesize sufficient amounts of arginine, cysteine, glutamine, proline, selenocysteine, serine, and tyrosine due to illness.